Chapter 17, Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by the organism to indirectly break down simple molecules like glucose, amino acid, fatty acids, etc. Now, when we add oxygen with glucose, we get ATP, and that is the energy which we get to perform various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reaction. It is therefore evident that O2 has to be continuously provided to the cells, and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmospheric with the CO2 produced is called breathing, or commonly known as respiration. Moving on to the next topic, that is the very first topic, the respiratory organs. Mechanisms of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelentrates, flatworms, etc. exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms have moist cuticle, and insects have a network of a tube which is called the tracheal tube to transport atmospheric air within their body. Specialized vascular structure like gills, which is known as branchial respiration, are used among most of the aquatic arthropods and mollusks, whereas vascularized bag called lungs is pulmonary respiration, which is used by terrestrial forms and for us also, for humans, for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds and mammals respire through pulmonary respiration, that is our lungs. Amphibians like frogs can respire through their moist skin, which is cutaneous respiration. Also, so what is vascularized tube? That is actually the tracheal tube, and then we have a vascularized structure called the gills also, which is known as a branchial respiration. Vascularized sac is known as the lungs, which is the pulmonary respiration. Now, and then moving on to the second subtopic, that is the human respiratory system. We have a pair of external nostrils opening out to the above upper lips. It leads to the nasal chamber through the nasal passage. Now, from the nasal chamber, it opens into the pharynx, a portion which is common to the passage of food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps the sound production, and hence it is called sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called the epiglottis. So epiglottis is actually the cover which can prevent the entry of food into the lungs. Now, which divides at the level of fifth thoracic vertebrae. So trachea is actually a straight tube extending from the mid-thoracic cavity, which divides at the level of the fifth thoracic vertebrae into left and right bronchi. Each bronchi undergoes repeated division to form secondary and tertiary bronchi, ending up in a very thin terminal bronchioles. The trachea, primary, secondary and tertiary bronchi and initial bronchi are supported by incomplete cartilaginous rings. Each terminal bronchi give rise to a number of thin, irregular wall and vascularized bag-like structures called alveoli. The branching network of bronchi and bronchioles and alveoli comprise the lungs. We have two lungs which are covered by a double-layered pleura with a pleural fluid between them. It reduces the friction on the lung surface. The outer pleural membrane is in close contact with the outer line, with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas from the alveoli Anther duct from the respiratory or exchange part of the respiratory system. The conducting part transports atmospheric air to the alveoli and clears it from foreign particles, humidifies and also brings air to the body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between the blood and the atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs on the lower side of a dome-shaped diaphragm. The anatomical setup of the lungs in the thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. Breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is released out. Diffusion of gases like CO2 and O2 takes place across the alveolar membrane. Transport of gases is by the blood. Diffusion of O2 and CO2 between blood and the tissues. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2, which is cellular respiration. And that is the end of topic 1. Moving on to the second topic, mechanism of breathing. Breathing involves 2 stages. Inspiration, during which the atmospheric air is drawn in, and expiration, in which the alveolar air is released out. This movement of air into and out of the lungs is carried out by pressure gradients between the lungs and atmosphere. Inspiration can occur if the pressure within the lung, that is the intrapulmonary pressure, is less than the atmospheric pressure. So if the atmospheric pressure is less, we breathe in, which is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place with intrapulmonary pressure, which is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles like external and internal intercostals between the ribs help in the generation of these gradients. Inspiration is initiated by contraction of the diaphragm, which increases the volume of the thoracic chamber in the anterior-posterior axis. The volume of the thorax chamber in the anterior-posterior axis, the contraction of external intercostal muscles lift up the ribs and the sternum. So as a result, there will be an increase in the volume of the thoracic chamber in the dorsoventral axis. So the overall increase in volume causes a similar increase in the pulmonary volume. So increase in the pulmonary volume, there will be a decrease in the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. So as the volume inside the lung increases, the pressure gradient will also increase. As a result, what happens is the volume increase. See, very simple, when the volume increase, the pressure inside will decrease. As a result, what will happen is there will be a pressure gradient between them, which is known as intrapulmonary pressure. So since this pressure is less than the atmospheric pressure, we breathe in. That is it about till here. That is about inspiration. Relaxation of the diaphragm and intercostal muscles returns the diaphragm and sternum to their normal position and reduce the thoracic volume, thereby the pulmonary volume. This leads to an increase in the intrapulmonary pressure, which is slightly above the atmospheric pressure, causing in breathe out, that is expiration. So we have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. So what happens is some of them have a good breathing difficulties. Good breathing ability. So not difficulty but ability. So this is given by the additional muscles which is present in the abdomen. On an average, a healthy human body can breathe 12 to 16 times a minute. The volume of air involved in breathing movements can be estimated by the spirometer, which is in a clinical assignment. That is not much important. And you need to remember that RBCs carry almost 99.5% of oxygen. Now I'll be giving a brief about, not introduction in the sense of summary about the above paragraph, since the words are very confusing. Giving a brief summary about what happened before, since the words are actually a little bit confusing when you read. So in short, inspiration, what happens is the lung volume increases. There will be a contraction of the intercostal muscles. As a result, the ribs and the sternum will lift up. So as a result, what happens is there will be an increase in the volume of the chamber, thoracic chamber, and there will be a increase in the pulmonary volume and decrease in the intrapulmonary pressure. As a result, what happens is, since the atmospheric pressure is more than the intrapulmonary pressure, air comes in. That is it about inspiration. Now, expiration is actually very simple. Volume increases, pressure increases. When the atmospheric pressure is less and the intrapulmonary pressure is very high, that means we can breathe out. So that is it about till here. Now moving on to 17.21, that is respiratory volume and their capacities. Tidal volume. Volume of air inspired or expired during a normal respiration, it is approximately 500 ml. A healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Inspiratory reserve volume is actually IRV. Additional volume of air a person can inspire by a forcible inspiration, and this averages 2,500 ml to 3,000 ml. Expiratory reserve volume, that is the ERV, is actually the additional volume of air a person can expire by a forcible expiration. This averages for about 1000 ml to 1100 ml. Residual volume, which is RV. Volume of air remaining in the lungs even after a forcible breathe out or expiration is called residual volume. And this is about 1100 ml to 1200 ml. By adding up a few respiratory volume described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity. Total volume of air a person can inspire after normal expiration. This includes tidal volume, inspiratory reserve volume. So, inspiratory capacity IC is equal to tidal volume plus inspiratory reserve volume, that is the IRV. Now, expiratory capacity is the total volume of air a person can breathe out after a normal expiration. That is tidal volume plus expiratory reserve volume. Functional residual capacity is the FRC, volume of air that will remain in their lungs after a normal expiration, which includes ERV plus RV, that is expiratory reserve volume plus the residual volume. So that is functional residual capacity. Now moving on to vital capacity, which is VC. The maximum volume of air a person can breathe in after a forced expiration. This includes expiratory reserve volume plus TV, that is the tidal volume plus the inspiratory reserve volume, or the maximum volume of a person can breathe out after a forced expiration. Total lung capacity is actually the sum of the vital capacity plus residual volume, or like you can add up everything. Residual volume plus the expiratory reserve volume plus the tidal volume plus the inspiratory reserve volume. That is it. So we are done with the terms. So we are moving on to the third topic, that is the exchange of gases, which is diffusion between the membrane. Alveoli are primary sites of exchange of gases. Exchange of gases are also occur between the blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on the pressure and the concentration gradient. Solubility of the gases as well as the thickness of the membranes involved in diffusion are also some of the important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gas is called the partial pressure, which is denoted by small p O2 for oxygen and small p CO2 for carbon dioxide. So partial pressure is the pressure contributed by an individual gas in mixture of gases is called partial pressure. Now, partial pressure of two gases in atmospheric air and the two sides of the diffusion is given in the table here. And the data indicates a concentration gradient for oxygen from alveoli to the blood and blood to the tissues. So, in this table it is given, this is important. See, respiratory gas O2, when the atmospheric air is 159 atm, that is the atmospheric pressure, what happens is the alveoli will be 104. So the deoxygenated blood will be 40, oxygenated blood will be 95 and tissues will be 40. When it comes to CO2, it is 0.3, 40, 45, 40 and 45. Now, a gradient is present for CO2 in the opposite direction from tissues to the blood and blood to the alveoli. As the solubility of CO2 is 20 to 25 times higher than O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in the partial pressure is much higher compared to that of oxygen. This diffusion membrane is made up of three layers. Now the very first layer is the thin squamous epithelium of the alveoli. Second is the endothelium of alveolar capillaries, and the third is the basement substance in between them. However, the total thickness is much less than a millimeter. Therefore, all the factors in our body is favorable for diffusion of oxygen from the alveoli to the tissues, that of CO2 from tissues to the alveoli. Topic number 17.4, that is the transport of gases. So blood is a medium of transport for O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of O2 is carried out in a dissolved state through the plasma. Nearly 20 to 25% of the CO2 is transported by RBCs, whereas 70% is carried as bicarbonate, which is HCO3. About 7% of CO2 is carried in dissolved state through plasma. So again summarizing, almost 97% of O2 is carried by RBC, remaining 3 is carried in a dissolved state in plasma, whereas in CO2 almost 20 to 25% is carried by RBC and almost 7% is carried by CO2. Moving on to the next subtopic, that is transport of oxygen. Hemoglobin is a red colored iron containing pigment present in RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2. Partial pressure of CO2, hydrogen ion concentration and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. And this curve is called the oxygen dissociation curve. So you can see a figure that is 17.5. It is actually, there is a bend when it comes to from 60 to 100, it bend and it reaches a constant state. So repeating again, what is oxygen dissociation curve? It is actually a sigmoid curve which is obtained when the percentage saturation of hemoglobin with O2 is plotted against partial pressure of O2. Now, hemoglobin is equal to 4 O2 molecules. That is very important to be remembered. Now, oxygen dissociation curve, and it is highly useful in studying the effective factors like PCO2, H plus concentration, etc. on binding of O2 with hemoglobin. In the alveoli where there is a high PO2 and low PCO2, lesser H plus concentration and lower temperature, these are the factors which are favorable for the formation of hemoglobin. I repeat again, high partial pressure of O2, low partial pressure of CO2 and less H plus concentration, low temperature leads to the formation of oxyhemoglobin in a reversible manner. Now, these conditions are favorable for dissociation of oxygen from hemoglobin too. This clearly indicates that O2 gets bound to the hemoglobin in the lung surface and gets dissociated with the tissues. Every 100 ml of oxygenated blood can deliver almost 5 ml of oxygen to the tissues under normal physiological conditions. Moving on to the transport of carbon dioxide. CO2 is carried by hemoglobin as carbaminohemoglobin, which is about 20 to 25%, and the rest of the 7% is in dissolved state carried out by plasma. This binding of the partial pressure of CO2 and PO2 is a major factor which could affect this binding. When PCO2 is higher and PO2 is low in the tissues, more binding of carbon dioxide occurs, whereas when PCO2 is less or PO2 is high in the alveoli, dissociation of CO2 from carbaminohemoglobin takes place. CO2, which is bound to hemoglobin from the tissues, is derived at the alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase, and minute quantities of the same is present in the plasma too. This enzyme facilitates the following reaction in both directions. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into the blood. So RBCs in plasma and forms HCO3- which is bicarbonate and H plus. At the alveolar site where PCO2 is low and the reaction proceeds in the opposite direction, leading to the formation of CO2 and H2O. Thus, CO2 is trapped as bicarbonate at the tissue level and transported to the alveoli, and it is released out as CO2. Now, every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. Giving a small summary because this seems to be very confusing and when you read it at the very first. So, when PO2 is low and PCO2 is high in the tissues, binding of oxygen takes place. If PCO2 is low and PO2 is high in the alveoli, there will be dissociation of CO2 from carbaminohemoglobin. That is it till here. Now, in the next paragraph, it is mentioned that at the tissue site, when partial pressure PCO2 is high due to catabolism, CO2 diffuses with RBCs and plasma, forming bicarbonate and H plus. No, at the alveolar site, what happens is usually PCO2 is low. The reaction proceeds in the opposite direction, leading to the formation of CO2 and H plus. That is it. And as a result, CO2 is trapped as bicarbonate at the tissue level. Now, every 100 ml of deoxygenated blood carries almost 4 ml of CO2 to the alveoli. Moving on to the topic number 5, regulation of respiration. So, human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system. A specialized center present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Now, another center in the pons region of the brain is called the pneumatotoxic center, which can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is present which is adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. So, increase in these substances can activate this center, which in turn can signal the rhythm center and make the necessary adjustments. in the respiratory process by which these substances can be eliminated. Receptors associated with the aortic arc and carotid artery can also recognize the changes in CO2 and H plus concentration and necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. Moving on to the last topic, that is disorders of respiratory system. First one, asthma, is the difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased, one of the major causes of cigarette smoking. Now the very last one, occupational respiratory disorder in certain industries, especially those involving